The Mendelian cross is named for Gregor Mendel, a 19th century Augustinian monk who performed and documented the first controlled genetic crosses and allowed humanity to begin to understand the nature of inheritance.
You're welcome.
A Mendelian cross begins with a controlled breeding experiment spanning at least 3 generations and is designed to determine the inheritance pattern of a trait. Now here's some terminology to help us describe these crosses. We call the outward appearance of a trait its phenotype. The phenotype of this flower is purple. Each version of a gene is called an allele. The examples in this video will have 2 types of alleles: dominant, represented by an uppercase letter, or recessive, represented by a lowercase letter. The combination of alleles an individual carries is called the genotype. Pea plants have 2 copies of each gene, so the genotype of each trait is represented by 2 alleles. The next concept to understand is that every Mendelian cross begins with true breeding parents. True breeding means that if 2 individuals of that variety of organism are mated, then all the progeny will resemble the parents for a particular trait. In this case, white-flowered parents always produce white-flowered progeny. Now, let's start the Mendelian cross. The initial parents in a Mendelian monohybrid cross will be true breeding individuals who differ from one another by a single trait, such as these pea plants, one of which has a purple flower and the other has a white flower. When it comes time to cross the 2 individuals, they'll be bred in a reciprocal cross. Now this means that pollen from the purple flower plant will be placed on the stigma of the white-flowered plant, making the mother in this cross white and the father purple. A second cross is also initiated where pollen is taken from the white-flowered plant and placed on the stigma of the purple-flowered plant, so the mother has purple flowers and the father white flowers. This type of cross is set up to ensure that the trait is truly inherited from both parents equally. Any progeny produced from the true breeding parents in a Mendelian cross are called the F1 progeny, or F1 generation. All the progeny in a cross like this will resemble one of the parents regardless of the parental phenotype. The parental variety seen amongst all the F1 progeny is considered the dominant trait. The parental variety that is absent in the F1 is the recessive trait. So for this cross, purple flower color is dominant and white is recessive. A Mendelian cross will continue for at least 1 more generation. For the next cross, 2 of the purple F1 progeny are chosen and crossed with one another. This means that the mother and father are actual siblings produced from the parental cross. Any progeny produced by this cross is called the F2 generation. In this generation, 3/4 of the progeny will have the dominant version of the trait and 1/4 will have the recessive, demonstrating that the recessive trait didn't disappear, but was hidden in the F1 generation. Let's start over and explain these patterns using chromosomes and alleles. Each individual has 2 alleles, or copies of each gene, and they reside on the homologous pairs of chromosomes that segregate during meiosis. For example, the parental purple flower plant will have 2 copies of the dominant gene, represented here by uppercase letter P, chosen to represent purple. This combination is called homozygous dominant. The white flower plant has 2 copies of the recessive gene, represented here by a lowercase p, chosen to represent a lack of purple pigment. This is called homozygous recessive. The F1 receives 1 allele or gene copy on a chromosome from each parent. This combination of 1 dominant and 1 recessive in the F1 progeny is called heterozygous. The F1 heterozygotes in this cross will appear dominant because they received 1 dominant allele from the homozygous dominant parent. In the F1 cross, both parents are heterozygous, so progeny have the possibility to be homozygous dominant, homozygous recessive, or heterozygous by 2 different combinations of alleles. The homozygous dominant and heterozygous progeny will express the dominant trait, and the homozygous recessive will appear recessive. These combinations of